Boa tarde a todos. Acredito que nós já estamos online. E hoje é um grande prazer representar um grande amigo da Universidade do Texas, no MD Anderson Cancer Center. Então vamos mudar para o inglês para poder apresentar o Dr. Roberto Rangel. So Roberto, thank you so much for joining us today. Roberto is just coming back from his vacations last week, so he has a pile of things to go over, but he was kind enough to accept our invitation to give this fascinating talk about reversing the immune-excluded, the cold tumor immune microenvironment in oral squamous cell carcinomas. So Roberto graduated as a chemist in Mexico, followed by a master's degree, a PhD, and a postdoc in immunology, both in Mexico and at the MD Anderson Cancer Center. Roberto was trained by very talented scientists, such as the Dr. Takmak at Amgen in Canada, followed by Dr. Renata Pasqualini and Wadi Arab with lots of different areas, including a way to identify and to target organelles that we have inside the cells using his iPhage technology, which is absolutely fascinating. And then later on, he moved to the Nancy Jenkins and Neil Copeland Laboratory at the Houston Methodist Research Institute. But following that time over there, very productive as always, Roberto was recruited back to the MD Anderson Cancer Center, going to the immunology department, and now he's assistant professor at the head and neck department. Roberto is a very close friend, a very dear, very nice person, a fascinating scientist. And when I was going to move to Houston back in 2006, we were already friends. And Roberto was known as the king of cloning, a man that can clone anything that you want. And also, I have to point that he makes the best fajitas in Houston. So, Roberto, thank you so much. The stage is yours. People are going to send eventually some questions here, and I'll pass them later on to you. So, thank you so much, my friend. It's great to see you again, and we're looking forward to our presentation.
Okay. Thank you for the kind introduction. And, I'm happy to participate and always willing to help people. So, today I'm going to talk, I have been working in different fields. And, when I joined this department, one of the major, challenges for us was, patients who have oral squamous cell carcinoma, the death rate is very high. Patients who come with very advanced stage, there's no good treatments for them. So we wanted to plan, how we can develop new models, new technology, or what kind of mouse models. So first, I'm going to provide a brief introduction. I'm going to provide some of the things that we have developed in our lab, how to approach this challenge, and some of the future goals that we're planning to move ahead. So just going to give a brief introduction about, worldwide head and neck squamous cell carcinoma, which includes oral cancer, is the sixth leading cancer and accounts over 600,000 new cancer cases each year. And as you can see in this graph, the frequency has been dropping since, tobacco was kind of more prohibited in certain places in the United States, but it continually is dropping, but still it's a very steady state, is still, some consequences. So what I want to show in this slide is that, oral cancer or head and neck squamous carcinoma, which includes oral cancer, it develops in an area that you have a lot of functions. I mean, you need to swallow, you need to breathe, and you need to be very careful when you provide treatment to these patients because these areas are very sensitive for all our functions. I mean, you have here the taste, the smell, the hearing, you have a lot of very important organs that are necessary for a physiological and to function well. You can see that approximately half of the newly diagnosed patients will not survive beyond 5 years. And, you can see here in the pictures how aggressive this can be in some of these cancers. Some of the factors that, cause this oral cancer is alcohol, tobacco, smoke, some better liquid, arecanut, some airborne pollutants. And, if you're exposed to this continuously, you tend to develop, some mutations. And it has been identified some mutations like P53, deletions of certain specific genes, or amplifications, or PI3K kinase. Some of the treatments, it's surgery. Of course, is going to depend at what stage the cancer is. Could be a minimal invasive surgery, and it can go to school-based surgery or different types of surgery. Also radiotherapy, chemotherapy, the usual cisplatin, docetaxel, carboplatin, or you can do a combination of chemotherapy and radiation. It has been introduced to increase the control to preserve the organ integrity. And of course, immunotherapy. Now, here, immunotherapy, I think 90% of MD Anderson is working in immunotherapy. And actually, the Nobel Prize for, I think, I forgot the year, but it's very recent, by Dr. James Allison, who is the chairman of the department. Now he has his own institute, Allison, of research. And he was the first one to publish a development of immunotherapy, how you can reverse, how you can wake up the immune system to destroy tumors. And another fact about this is that only 10 to 15% of patients with advanced head and neck squamous carcinoma will respond to immunotherapy. So this suggests that it's very poor response, and so what we can do. Another complications after the treatment is cosmetics, disfigure of the face, head and neck, difficult swallowing, dry mouth, altered taste, increase of risk of infections, and suicide. This is very interesting because you can see suicide in male pancreatic cancer patients is very high, but the second cause is head and neck cancer patients. So these are real concern. Sometimes patients, they don't die about the real primary tumor or cancer, is because the way of life, or they have complications like swallowing, breathing, or other causes. So the clinical, histological, and molecular pathogenesis is, you have here your normal mucosa, start with the level hyperplasia, and it has been detected that some of the mutations like P16 inactivation of loss of function, actually P16 is located in chromosome 9, it's a loss of heterozygosity. Then you can move to dysplasia, carcinoma in situ, and carcinoma. And depending on each stage, you're accumulating more mutations, amplifications of oncogenes, or inactivation of tumor suppressors like P10. So, with the advance of technologies and next generation sequencing, the TCGA has developed this kind of very nice map showing you how, receptor tyrosine kinases can be altered in head and neck in HPV negatives, patients. Oncogenes, some of them are amplified. Some of these genes like PI3 kinase, P10, some of these tumor suppressors are inactivated, or some are amplified. However, one of the tumor suppressors that is mostly altered is P53 in 84% of the cases. This is really impressive, to have this kind. And the second one is P16, 58%. And most of the times is, some of them are mutations, most of them are kind of homozygous deletions. And P53, you can have the loss of function, that is the inactivation of the gene, or you can gain a mutation. For example, here, P53, as I mentioned, is the most frequent mutated gene in head and neck. And most of the mutations are on the DNA binding domain of the protein. And these are the hotspots, R175, 248, 245, specific amino acids. And you can see the arginines are predominant. To change these amino acids, then this protein becomes a gain of function. Now, you can see here in this cartoon how P53 regulates a lot of functions. Amine regulates DNA damage response, autophagy, cell cycle, metabolism, almost anything that you have a function in the cell relates to P53. But little is known about the role on the immune system. There are some papers showing about inflammation in the tumor microenvironment, how p53 modulates some of these factors that are relevant. So why I'm bringing this? So, uh, in tumors, when I studied cancer biology, that was probably a long time ago, uh, we only knew about oncogenes, and then the wave of angiogenesis with the vasculature, then come with the tumor suppressors. But now there's, after 20 years ago, and start moving the field more to explore what are the components inside of the tumor. And people found a lot of soluble factors, and these are cytokines that they have an effect on immune cells. I mean, I remember when I was taking my classes in immunology in Mexico, and I asked why the immune system doesn't destroy tumors. And the professor told me, no, it's because they don't express antigen and the cell don't go inside. So that was the one sentence explanation. But now things are changing, that we know that there are immune checkpoints inhibitors. These immune checkpoints are molecules that are the brakes of the immune response. For example, when you have a flu, fever, you have a very strong immune response, but at the same time, it gets to a peak, and all these molecules are being expressed in the immune cells. So the endothelial cells and everything start expressing all these molecules to stop or make the immune cells like a more exhausted or energetic. So gradually, they are decreasing the immune response. Now, tumor cells, they learn how to express these immune checkpoints. They're able to put the brakes on the immune system. And not only that, they can recruit immune cells, for example, like Tregs. These are regulatory T cells that they can secrete factors and inactivate immune T cells. Or also tumor-associated macrophages, TAMs. They also secrete cytokines, and they can inhibit the activation of immune cells. So the tumor, in a way, learn how to protect and how to survive the immune system. So now, how we can study this? I mean, this is very nice. We have these systems, and what kind of models, we can use. So there are some models, system uses for tumor biology. The one on the top, you have a human patient where you get a tumor, you put it in culture, and you're going to generate a human cancer cell line. But you use an immunodeficient mouse. So it can grow there, but this mouse doesn't contain the immune system. Now, you can use a patient-derived xenograft model into a mouse, but still, it doesn't contain all the components that you need to study how it's fighting tumor cells against the immune system. Now, there are two systems that are very interesting. One is the Syngenic Models, that they are pure mice, that they contain an immune system, immune-competent mice, and they contain all the components. So, for example, you get a tumor in a mouse, Syngenic mice, you put it in culture, and you put it back in the same mouse, same mouse strain, and then you have all the components. You have the immune cells, the fibroblasts, the vascular network, and the tumor cells. Now, if you want to add a little bit, become more fancy, you can use genetically engineered mouse models that I have been working for the last 20 years, using this type of models. So if you can combine these two technologies, it's very powerful. So now what we have available. So when I got here to the department, there were only 2 cell lines that there were available for the whole community. We're doing oral cancer for, trying to understand immunotherapy. It was MOC1 and MOC2. These cell lines were generated exposing mice with DMBA carcinogen for several weeks. Mice developed tongue tumors, and they were able to generate and immortalize those cells. However, these cells were generated by Dr. Upalori, and actually we have the cells here in the lab, but they don't resemble exactly what it really happens with the tobacco signature. They have a mutation like HRAS that is not present in patients with cancer. So, it's a good model. That's what we have at that point. Then recently in 2000, I think by the end of 2020, Dr. Silvio Gatkin, he published a very nice paper generating 4 cell lines. 2 of them are tumorigenic, And we have this model. So these mice are coming from wild-type mice. They acquire a mutation in p53, and they're able to grow and become immortal. So I studied during this time. I was working to establish this model and characterize exactly the time points, when you can develop and try to understand if this carcinogenesis model can resemble human pathology. And yes, so when you expose mice with carcinogen in the water, drinking water, we use C57 black. And the reason we use C57 black mice is because it's one of the mouse strains that has been mostly used for more scientists who work in immunology. There's a lot of tools, and so we expose mice with foreign QO treatment for 16 weeks. So this means like 4 months. So, for graduate students, it's a long time. Between 12 and 16 weeks, you start observing mild dysplasia, moderate dysplasia, and severe dysplasia. After you stop the treatment, you let the mice for additional weeks, between week 20 and 24, and you start observing squamous cell carcinoma. So this is 6 months plus, the processing and the quantification. These experiments are long, but you can study premalignant lesions and you can study squamous cell carcinoma. So it's a system that is very flexible, and it shows all the pathological characteristics what happens in humans. Now we have established this model and well characterized. In the meantime, I had some mice that I ordered. These mice are commercially available here in the United States, and we crossed them with a K14 transgenic mouse. So what it means here is we have a wild-type mouse that will be our control. We have another mouse that contains p53, loss of function. So I want to mention here, K14, is a keratin 14 promoter that drives the expression of CRE. CRE is going to delete or inactivate p53, but only on the stratified epithelium. This means on the skin or in the oral cavity. Now, and this other mouse contains a gain of function. So we have two types of mice, one that is gain of function, one that is loss of function. And we selected p53 because p53 is 84% of people who has organ carcinoma, they have these mutations. These mice, were crossed. Actually, I back-crossed these mice because we obtained them. They were not 100% black 6. So it still took me 1 year more to back-cross these mice to have it in a C57 and then start the experiment with foreign QO. So we have here combination of Syngenic model with a genetically engineered mouse model. So the strategy was to develop tumors, put it in culture, immortalize, and use them as a platform or a model for our purpose. So here, this is just a diagram how we have our mice, or genetically engineered mouse models. These are the different tumors that we collected from the tongue. So we call it ROC1, ROC2, and ROC3. These are the name of the cell lines. ROC1 comes from a mouse, p53 wild type, ROC2 from a knockout, and ROC3 from a gain of function. So we obtain the tumors, half of the tumor we send it for histology, the other half we put it in culture. So we put it in culture, and then we did a flow sort for EGFR positive cells. So we know that most of the oral cancer cells, they highly express EGFR. So this was done with the purpose to remove any fibroblast contamination, enrich with the soft agar. We grow colonies, we pick those colonies, and we grow the cells. So after we have all these cultures, so this just single slide is 1 year of work. We next move to see if these cells were tumorigenic. So after the cells were immortalized, we went back and inject them in the tongue of the mice. So these are tongues that were injected with different concentrations of cells, going from 500,000 to 10,000. ROC1 grows from, even 10,000 cells, they can grow to 500,000. ROC2 and ROC3, you can see a very nice grow, 500,000 cells, but ROC2 doesn't grow 100. 100,000 cells. So this is, they do not grow. And this ROC3 also grows 500,000, but at 100,000, you can see start growing, start shrinking. But we try to keep mice for a long time, and here you can see even here for 80 days, we start observing tumors. So there's something happening here that we're trying to explore. What will be the function? What is happening during this period of time with this amount of cells? And another thing is some of these cell lines generate metastases. That is one of the things, patients die because of metastasis. ROC1 is the most metastatic cell line, showing, very high frequency. So we already have these cells generated with different mutations. However, I just want to mention that the mice were exposed with a carcinogen. So we need to explore what other type of mutations were acquired with the carcinogen, not only p53. So we went and to do next generation sequencing. We did whole exome sequencing from the 3 cell lines, and then we compare the most frequent mutations observed in humans with in the mouse. So these are from TCGA. So ROC1 that came from a wild type mouse acquired a mutation in p53. It acquired a T122 mutation, so it was no more anymore wild type. ROC2, of course, it contains a deletion, was confirmed, and only acquired a slight mutation here in one of these genes. But ROC3 is the cell line that contains more mutations associated with head and neck squamous cell carcinoma. So we have characterized these cell lines, and we explore a little bit more just to see if they acquire kind of the signature of foreign QO, that is C2A and C2T. And yes, we observed that C2T, the 3 cell lines contains, however, C2A, ROC3 contain many more. The frequency was very high. Also, ROC3 shows that there's an increase of frame shift deletions. This was totally unexpected, suggesting that probably this gain of function make the cells more genomically more unstable, and thus perhaps there are more mutations in these tumors. So after we have characterized, the mutations, confirmed that we don't have any more a wild type, tumor. Now we have a acquired mutation. So ROC1, ROC2, and ROC3, the 3 of them contains p53 alterations. So the next step was, try to characterize. And this is just a cartoon showing that what is a cold tumor and what is a hot tumor. I mean, a cold tumor excludes CD8 T cells, NK cells from the tumor. It contains infiltration of Tregs, and is a poor prognosis and response to immunotherapy. Hot tumor, it's when you have a high infiltration of CD8 and natural killer cells. You have suppressions of different types of cells, and you can improve the killing by using antibodies to block these immune checkpoints. Like PD-1 is a molecule that is very commonly used, so you can come with an antibody, block this PD-1 from the T cell, and then you can destroy the tumor. So these are very general characteristics. So the next step was to characterize the tumor immune microenvironment of the different ROC cell lines. So we inject the cells again in the tongue. We did some staining, immunohistochemistry, and quantification. Now, ROC1 is the one that is excluding T cells. So this suggests that it's a cold tumor compared to ROC2 and ROC3, that they have a high infiltration of CD4 and CD8 T cells. Also, there's an increased infiltration of Tregs in ROC2 and ROC3. And interestingly, we observe a high infiltration of dendritic cells only in ROC3. Dendritic cells are the masters of antigen presentation, and ROC3 is the one that contains more frame shift deletions or frame shift alterations. So this might suggest that perhaps ROC3 generates new antigens, and these are eliciting the the infiltration of dendritic cells. However, if you have infiltration of these cells, you need to have cells to counterattack the immune response. You have Tregs but also you have a lot of tumor-associated macrophages, M2 and ROC3, like I'm showing here. It's tumor-associated macrophages. M2 are masters of secreting TGF-beta and inhibiting the activation of dendritic cells or in immune cells. In addition to that, there's a high expression of PD-1 immune checkpoint inhibitors and ROC2 and ROC3 that are warm tumors, as well another immune checkpoint TIGIT. So this strongly suggests that, in summary, ROC1 is a cold tumor that contains more immune-excluded and less immune checkpoint inhibitors compared to ROC2 and ROC3. So then here we profile a large list just to identify what other immune checkpoints or targets could be available. So the next step was, all right, so we have generated this mouse model, we have characterized the immune microenvironment, how we can use it. So we know that PD-1 is being used a lot in head and neck cancer patients, but also there's a new target called TIGIT, and other groups in here in MD Anderson, they're using STING agonists. In this way, I want to mention PD-1 is an antibody that will block PD-1 or an antibody that blocking TIGIT. STING agonists, these are dinucleotides that they go inside the cell, activate the pathway of the interferons, and the interferon secretes all these factors to activate the immune response. So they can act dendritic cells, antigen-presenting cells, CD8 cells. So these can make our cold tumor very warm and, promote infiltration and the destruction of the tumor. So the first one we test is the ROC3 tumors. The ROC3 tumors are hot tumors that have immune cells. So we inject the cells, and then at day 5, we start with the different immunotherapy treatments using the antibody, anti-PD-1, or TIGIT. In panel B, this is the tumor growth of mice treated just with the isotype control. This is a normal antibody that doesn't target anything, it's our control. So tumors are growing, they don't respond. When we use PD-1, there was an immediate response in mice, tumors regress totally with PD-1 antibody. We keep the mice for 80 days. They did not, form any tumor. Even we rechallenged these mice at day 90 with more ROC3 cells. The tumors did not grow. So this strongly suggests that we generate immune cell memory. This is a hot tumor, and you just need to block one of these immune checkpoints. With TIGIT, there were only 2 tumors that they resist the therapy. So this is very normal to observe when you do immunotherapy treatment. Not all the tumors going to respond, but 80% of the tumor responds. So at this point, we know that our antibodies, they work, they're efficient. We have a model that you can, it responds to immunotherapy. And also, we test the STING agonists. These are small dinucleotides that we did, we did intratumoral injections. So after 3 doses, tumors disappear and they have a very good response. However, as you know, this is very nice. You see this response is great. However, we want the challenge that we proposed at the beginning. Why patients are being resistant. That's the main issue. So we selected one of the tumors that was a cold tumor, the ROC1. We did the same experiment. We did the injections into the oral cavity. We did different treatment with the antibodies, and none of them respond. Even we tried to do a combination. The mice developed tumors. This tumor was resistant to immunotherapy. It's a cold tumor. In addition, we used the STING agonist, the 3 treatments. Tumors tend to shrink, but after a few days, they start going back. So this was kind of surprising, and interesting, because, it is nice to see positive results, but also it's good to have a model that is resistant, because it will allow you to study a little bit more what might be the mechanisms or how you can overcome this kind of resistance. So at this point, we have developed our mouse model. We treated with therapy, and we know that we have a model that is sensitive to immunotherapy and one that is resistant. Now, the next part was, okay, So how P53, because we know P53 is one of the main, genetic drivers in head and neck squamous cell carcinoma, and how it's playing. And there's some things in the literature that, regulates angiogenesis, regulates migration invasion, extracellular remodeling, and also some of them suggest that there's some regulation of secreting factors for infiltration of immune cells. So the question was, how mutant P53 modulates immunosuppression mechanisms, and how we can reverse this cold immune-excluded microenvironment. So what we did is, the ROC1 cell line, we inactivated P53, we knocked down, which are here by Western blot, and then we inject the cells in black 6, the same mice that we're using. I mean, your parental, your control, your scrambled sRNA, but the P53 knockdown did not grow. However, when you use an immunodeficient, a mouse that doesn't contain an immune system, mature T or B cells, the tumors grow. There's a delay of growth, but tumors grow. So this strongly suggests that the P53 was modulating factors or something that it allowed, to prevent the activation of the innate and adaptive immune system. So next, what we did is we took the same cells, the control and the knockdown, and we did Rna-Seq. So when we did Rna-Seq from the ROC cell lines, we observed that there's a big change on cytokines. And cytokines are very important for the attraction of immune cells and maturation or immunosuppression. But most of these cytokines that they are upregulated when you knock down, they favor the infiltration of immune cells. However, some of these, for example, IL-33, IL-16, or some of these cytokines are important for immunosuppression. So these Rna-Seq, we confirm all these findings by qPCR. And then the next step, we did the RNA, then we did a protein level. So we took out the supernatants and sent them for ELISA. So we did an antibody array, and we quantified, the full increase of cytokines when you inactivate P53. What it was surprising for us was this pro-MMPP that is highly expressed when you inactivate P53. These metalloproteases are very important to degrade extracellular matrix. This might favor infiltration of immune cells and destroy the tumor cells. There's an increase of IL-6. Now, IL-6 is, I want to say, is a very moody cytokine. Some people say that is important for immunosuppression. Others say that is important for to activate the immune system. However, until now we have not found the means how to locate the gradients of these cytokines and where are located in the tumor. That will bring us more feedback. So in this cartoon, I just want to show here that the green arrow shows the cytokines that are being, for example, affected when you do mutant P53. For example, when you inactivate this mutant P53, all these cytokines are going to go down. So you don't have M2 macrophages, but you have all these cytokines that are being upregulated that will favor M2, M1. And as well, the activation of CD8s, inhibiting of Tregs. So everything is changing the microenvironment. So in order to validate if these cytokines that are being secreted have an effect, in real cells. So we obtained, again, black 6 mice. We did a bone marrow flushing. So where you have most of them, all the bone marrow progenitors, and most of these you can differentiate it to myeloid cells, and you can see and then mature what type of macrophages. So we used two conditioned media. We used the control media and P53 knockout media. We exposed the bone marrow progenitor cells, and then we did flow cytometry. And we observed that when you have a knockdown of P53, the supernatant induces M1 macrophages, that they are very important to destroy tumor cells. In contrast, when you have knockdown, you have less M2, that this suggests that it's polarizing or cytokines. When you inactivate P53, you generate cytokines that will benefit and prevent the formation of cells that will favor tumor growth. So at this point, we have kind of confirmed the role of p53 in the environment. So next, we wanted to identify these, what kind of vulnerabilities we can find in this immune-excluded. And how do you remember when we did the experiment with the STING agonist? The tumors grow, we give it 3 times. It was a point where the tumors start, stop growing, start shrinking, but they came back. So we'd repeat the experiment. We obtained the tumors at these specific times. We did some slides, and then we tried to analyze what kind of molecules or immune checkpoints are being expressed, or what type of cells are being infiltrated during this period of time of response. So we did an open multiplex that allow you to stain for several antibodies, up to 6 antibodies. So the first experiment here, we did immunohistochemistry, was to show here these are control, but we observed an increase of expression of STING, that is the protein that responds for STING agonists, of course. But we observed a high infiltration of cytotoxic CD8 T cells and a reduction of Tregs at that window time with the treatment. Also, there was a reduction of M2 macrophages. However, when we analyzed for CD8 T cells, we analyzed for the expression of immune checkpoints, and there was an increase of CD8 T cells, but they were expressing the PD-1. So the strategy, now that we identified that PD-1 was highly expressed on the CD8 T cells, then we went back and did a combination therapy with STING agonist and immunotherapy, that is PD-1. So now that the tumors that, they start growing, the one in black treated with only with PBS, they start growing. But when you do a combination, tumors shrink. Of course, 2 tumors did not respond. So 80% of the tumors responds to the combination therapy, and this allows us to extend the life of the mice. So currently, we're exploring a little bit more from those 2 slides. I still have some I sent to analyze for a more comprehensive analysis, and we came back with multiple immune checkpoints and a lot of new molecules that I'm planning to continue to validate. And probably that will be another story to tell later in the future. And with this, I just want to summarize that we have generated and characterized syngenic oral cancer cell lines. We have identified their mutation landscape by whole exome sequencing. Also, we have characterized a tumor immune microenvironment and identified what is the role of this mutation of p53 in the tumor microenvironment. And we have established these new mouse models that can be very useful, for more future studies. And currently, because we have p53 mutations, I have another mice that is p16 that I'm following the same studies. And I'm doing a combination of the 2 mutations of p53 and p16, because we know that patients with head and neck or oral cancer that contains the 2 mutations, a deletion of p16 and p53, they do the worst prognosis. So we're developing new reagents. We have all the mouse models, and of course, looking for funding, to do all these great experiments. So I just want to acknowledge the people from MD Anderson, and I want to show that Jewen, she was my first graduate student. She came to my lab, and I can say she did most of the 90% of the work that I present. She's Jewen. She's the one holding the rat ear. And actually, this is a picture that we took in February 2020, just before COVID hit us in March, and we had closed our institution, and I was mouse work for 3 months. It was very catastrophic on my side. Bing Wang was a research technician. Yeah, she already retired. Tom, he helped us a lot with the pathological analysis and feedback with immunohistochemistry. Ron Wang and UK, there are 2 fellows that did research, and they were at the beginning establishing test rock cell lines. Fred, and Curtis are these two guys who helped me with all the whole exome sequencing and bioinformatics, as well the Department of Bioinformatics with June and Jing. And Abdullah, he has been very nice guy, giving support and different experiments. And Andrew G. Securata is an immunologist here in the department. And of course, Dr. Myers, who provide all the funding, the support, and the one who hired me in the department. So I just want to say thank you to all, and thanks for your attention.
Hi, Roberto. That was a wonderful presentation. We really enjoyed how you're moving your career and developing these models and all these cell lines and bringing key and very timely questions for us and for the audience. And also, it was good to see Fred's name in your slide. He was, as you know, he did his PhD here with us, and
Yeah.
we're glad to see that he's also moving to new paths and new careers. We have a few questions here. First, from Dr. Tiago Goss.
Mhm.
Tiago is a researcher here at the A.C.Camargo Cancer Center, and he sent me a question here by WhatsApp. So let me go there.
Mhm.
Uh, he's asking, Emmanuel, can you ask him? That was a very cool presentation. I would like to know if he did some experiments or some comparisons between your cold tumors, the ROC1, and the cold and hot tumors in humans. And he said that he understands that ROC2 and ROC3 are more immunogenic, and maybe this can explain the fact that they grow well in animals that have been inoculated. He would like to know if he did some of these inoculations in immunodeficient animals as well, and also about these comparisons of eventually whole exome sequencing and RNA-Seq between cold human tumors and cold uh, tumors after these cells.
Yes.
And Tiago is going to add something more. He's said that to acknowledge you that you gave a very exciting presentation.
Okay. Thank you. So, well, we're preparing another manuscript, another paper, actually comparing these the cold tumors that we have and the mutation of p53. They correlate in human patients, and they correlate in immune cell infiltration, and they correlate based on the mutation of p53. Whenever we see, when you have these kind of mutations of p53, you see more in gain of function, you see less immune infiltrations. Um, Now, regarding the other part of the question was, we have injected all the cell lines. All the cell lines, they grow in immunodeficient mice. I didn't show that data.
Mhm.
The ROC1, ROC2, and ROC3, they grow up even at very low dose. But when you have high, in immunocompetent mice, that's very interesting. Uh, the ROC3 cell line that contains more frame shift deletions, when you inject a 100,000 cells, they start growing and then start shrinking and then start going back again. So what we are reading right now, we are obtaining the tumors after the curve and select that population of cells and see if there's a difference, if what kind of new antigens. We're trying to analyze also all these frame shift mutations, exonic mutations, and see if they make a new neopeptides that can be presented to activate immune cells. So we are trying to follow all these kind of avenues on the rock cell lines. And now you know where the R comes from. Yes, from the rock cell lines. So um, so yes, we are continuing to explore all these. And what is interesting is, and I want to make this, I didn't discuss in the presentation, is that all these mutations, for example, the ROC2 and the ROC3, these are coming from genetically engineered mouse models. They already have a mutation in their germline. So these mice, they already have genomic instability even before foreign QO. Okay. And these are the two hot tumors. So this shows that at very early stage, even before foreign QO, probably there's a attack during the immune system to try to develop an equilibrium, between both of them. Just counter attack. the ROC1 is a cold tumor. This was coming from a wild type P53. This mutation was acquired during carcinogenesis. And this mutation needs to be very good to select and become very aggressive to survive, the immune response. So it's interesting that the mutation that was acquired with the carcinogen, okay, becomes the more aggressive. And the two mutations from genetically engineered mouse models that were in the germline, they are more warm tumors. So there might be some differences in there, in the system, but that's what we have. And currently we're trying to do the P16, the CDKN2A mutation that is, also 54% of the patients contains this deletion. And also we're doing double mutants, P16 with 172, 245 loss of function. It's a lot of mouse work.
But I know how you work. Kind of a machine in making these models. I have a few questions as well, Roberto. Have you looked for mutation signatures
Mhm.
in ROC1, ROC2, ROC3? What have you seen? Do you see a mismatch repair signature, for instance?
Yes, there are some. I've not had to explore a lot. We compare, the head and neck most frequent mutations, but there's a lot of mutations on genes on the PI3 kinase, on this tumor suppressor pathway. But in mismatch repair, we have found some like BRCA1, in some a mutation there. In another one, I have checked other ones I didn't observe, but it's very low in mismatch repair.
Yeah, because I think it would be interesting to look for mutation signatures
Yes.
when you see a kind of a specific pattern or of genomic alterations that would suggest a specific pathway that is active
Mhm.
in those cells and eventually triggering different phenotypes. Dr. Israel Tojal here at A.C.Camargo, he developed a very nice bioinformatics pipeline for
Okay.
identifying specific signatures. Maybe we can talk about it later if you're
Yeah,
interested.
one
But
of the
it's
things,
very informative. Yeah.
yes, one of the things is, here we want a lot of people from bioinformatics.
Yeah.
It's the people that is very busy. I had a lot of questions, but the only things that they can provide me were, these kind of things. But there's a lot of things. I have RNA-seq data from this, I didn't show, but I did all the analysis by myself, with the tools we have in the
Yeah.
internet.
Not easy. Yeah.
Not easy, but if I had someone that really analyzed all the RNA-seq data, probably they can find a lot of pathways and signatures. Yeah.
Yeah. Of course. And how frequent is this T122N that you saw that was a new acquired mutation?
Yes.
How frequent is that in humans?
That
And does it correlate with something?
well, that's a good question. Interestingly, this mutation is also found in another MOX cell line, but with a different amino acid. It's T122 a proline.
Hmm.
There's another group in Taiwan who did the same mouse work, and they did the same mutation, T122N, and it was very aggressive. But it was not high, they just show, metastasis, whatever I did, but they didn't explore the immune microenvironment. But in humans, this mutation, it's not detected, in head and neck squamous cell carcinoma. It's been detected in skin cancer, in P53, exposed by UV. And one of the things that we were planning to do is to make this mutation and overexpress it in human cell lines in head and neck, and see if they're modulating the same things that they observe in the mouse. You know, but the reviewers will say, well, why are you going to generate this mutation if not that frequent in humans? But I mean, it will give you an answer of your biological, your question.
Very, very nice. That's really intriguing.
Yes.
Um, Also, did you check for metastasis?
Mhm.
Because you show that metastases are only present in
One.
rock one,
Yes.
exactly. And what happens to the metastases in the other ones as soon as you start modulating the mutation profile, and you see the cytokines. When the tumors are responding, and those that do not respond later on, is metastasis varying a little bit?
Yes, we have not explored yet, the metastasis. With, of course, I mean, the tumors do not grow when you inactivate p53, but we have not observed when we treat the mice with these immune checkpoints and all, with the combinations. We have not explored yet. There are some people in the lab that are very interested to explore at different stages. We have labeled all these cells with GFP just to track and luciferase, just to see. The issue is because when we inject the cells in the tongue, the tumors are zero 0.5 centimeters. So if they go above 0.5 centimeters, the mice cannot eat. So the mice start losing weight. So there's no way to follow up a long study.
Oh, yeah.
So it's very short window that you have in there. So we're planning to generate like an inducible system to induce the inactivation of p53 a certain time with doxycycline, and then turn it on back, or or do a surgery in the tongue. Maybe I asked Dr. Myers to do a surgery there, but it's they're very interesting things, but we have not explored yet metastasis.
Yeah, and also I have my final question. Then there's another one from Dr. Tiago Medina, and you showed very nice charts where in one of them, for instance, 80% of the tumors responded very well,
Yes.
but 20% of them were still there, and
Mhm.
some of them after some reduction, and then they came up growing again.
Yes.
Please tell me what you did with these. Have you sequenced the exome or the RNA-seq in those that are not responding? Are these new clones? Did they acquire the new mutation? What happened to them?
Well, we tried to put them in culture, isolate them. We couldn't get, I think my student was very overwhelmed. She didn't took good care, but we tried, to get some of those tumors. We collect them, but we didn't follow up. Now
Okay.
I'm going back. Now I'm trying to make by myself, collect the tumors, put it in culture, and I'm the one that I'm supervising. So I generated other tumor cells derived from these resistant, immune checkpoints and other chemotherapeutic drugs. So that's kind of a more in future experiments ongoing.
Yeah, and Diana is asking here, saying that he has to do the single cell sequencing of those 20% that are growing, and of course, me, Diana, we love this
Yes.
genomic site, and we don't have to grow them again, but at least to see if you have new acquired mutations or what's going there. And let me go to Tiago Medina question. So Tiago is our chief immunologist here, if I may say. He's thanking you for the talk, and he's saying, well, STING is classically known to be phosphorylated by cGAS, which is a sensor of cytosolic double-stranded DNA,
Yeah.
which is commonly activated by virus infections.
Mhm.
So he's asking, mechanistically, how do you think that the STING agonists could be acting in this system and potentiating the anti-PD-1 response?
Well, we have not explored, mechanistically here. We have done some preliminary data, and we have checked the cGAS STING pathway stimulated with STING on the Roxo line. And yes, IRF3 gets phosphorylated. We have shown that cells start secreting more interferon alpha and beta, and those are the factors that are being activated. So we know that the pathway is being activated, but we have not explored, how to make this connection with the PD-1. There's many cytokines. For example, you have pro-MMP9 that is highly being secreted in the microenvironment. This destroys extracellular matrix. We don't know if this destruction of the extracellular matrix breaks down any gradient of cytokines or inflammatory cytokines, or allows the infiltration of immune cells. Now, the next part for us is to determine the infiltration of these immune cells are really specific for this to destroy the tumor, but we need to do in vitro experiment just to show, or is because there's an infiltration. So we're still, trying to explore with this. But I just want to say with STING agonists, it's not only for interferons. We have observed another immune checkpoints highly expressed, like BST2, LILRB3, LILRB4. There's a high expression of antigen presentation, MAC class 1. Did you notice preliminary data that I have? I'm analyzing currently, and I can share, but there's a high response, of a lot of things, and they're kind of gradually modulated because we did different time points when it goes down and then when it goes up. So we're finding a lot of things now. Tumors are very heterogeneous, so we want to do like a spatial transcriptomics, to localize, to have a map where it's happening, everything. And of course, I wanted to do single cell Rna-Seq, because that it will give you the power to know what cell is expressing what, in the microenvironment. So, but those experiments are expensive. So, I'm writing grants for applying for that, those experiments. So that's what we're doing right now.
Yeah, that's fascinating, Roberto. As you mentioned, MMP9, we have a question here by our dear friend, Professor Luis Giuliano. And he's asking, how do you see the room for the study of metalloproteases in your system?
Well, we have not, I used to work with CD13. It's an aminopeptide. You remember the knockout that they did for Renata and Wadi? But from there, I know it's a very interesting field, and, how they might play a role in remodeling the extracellular matrix or promoting angiogenesis and all these factors. And, I'm doing a phase display screening, using these models too. But I have not, I don't have enough people or, things. But, if people want to collaborate or, we can send the cell lines to you guys.
Yeah, Dr. Giuliano is an expert in metalloproteases here.
Okay.
Lovely, Roberto. I guess that we are running out of time.
Yes.
But it was fascinating. In the beginning of your talk, we had close to 200 people watching. So you draw a lot of attention as always. And I'm sure that everybody was fascinated about your brilliant presentation. So, my dear friend, thank you so much. It was great to see you again.
Yes.
Please send my best regards to everyone in the lab and in your family. And thank you so much.
Thank you. Thank you. Happy. So anytime, yes, invite me, let me know.
Absolutely. We invite you more again and for your new discovery.
Maybe São Paulo will be good.
Yeah, who knows? Maybe next time in person, yeah?
Okay.
We have the next Frontiers to Cure Cancer in 2023. So maybe it's going to be in person. We've been doing this virtually, but maybe we would love to meet you.
And good luck in Qatar.
Oh, thank you. Same to you. Bye-bye, Roberto.
Bye.
To you. Take care.